So one of the key events for our summit, as it was in 2018, is the final stage of the competition of the Magee Prize. And a few words about the Magee Prize. First, the $1 million that comprised the Magee Prize, that amount was donated by the Richard King Mellon Foundation, and you're going to hear much more about the Richard King Mellon Foundation throughout the evening at our dinner time. And we also have a little movie to detail the work that they've been contributing to. This is a prize that we established in our first summit 3 years ago, and it's designed to support the best research that we could identify for a period of about 2 to 3 years. There are 3 reasons why we're doing the Magee Prize. The first one, and perhaps the most important one, is to draw attention to the field of reproductive sciences, early human development, and women's health. A second reason is to try to catalyze transformational research, not just ordinary work, but research that can really transform the field, most innovative, even I would say disruptive field, what we'd like to call high risk, high reward type of research. Perhaps type of research that would not be funded by the most classical, traditional NIH or other funders. A third reason, which is really embedded within our the type of award we give, is the fact that we want it to be an international collaboration between somebody working here at our place here in Pittsburgh and somebody international. Now, as you'll see in a few minutes, all of the presenters are more than 2 people. Actually, the groups are 3 people, could be even more, but at least we want to have somebody working here and somebody working internationally, centering on any topic, basic, translation, or clinical, within the landscape of reproductive sciences, early human development, and women's health. So I'll say one word about the process. We've received a number of letters of intent and applications, and people asked us about it. And ultimately, we selected 15 applications that were suitable for review. And the first review phase was conducted by 4 professional, well-seasoned senior scientists who selected the best 6 applications out of the 15. And comments were given back for the ones that were still excellent, but there were 9 of them did not fit really our criteria for best proposal, best work. The 6 best applications were further reviewed by other scientists, not the initial 4, but another group of scientists. They're actually not only from here, but they're international, and they reviewed and gave us scores. And that led to the selection of the best 3, which all of us are going to vote today. So this has been our process. We've been quite pleased with how things worked, and we're quite pleased with the quality, as you'll see in a few minutes, of the 3 best presentations. So we're going to be listening to the 3 presentations. Each one will be 15 minutes. We're not going to take any questions because of time and pace, and we wanted to be fair to everybody. And then after they'll be presented alphabetically based on the PI's last name, after they'll be presented, then we will vote. And I'll talk about the voting process after we're done listening to the 3 presentations. And then we're going to end up voting based on the quality of the presentation, with 1 being the best score and 9 being the worst score. Like I said, there will be no time for questions. We'll go through the 3 presentations, we'll vote, and then we're going to take a break. We have terrific breakout sessions, several of them, that will be right outside. And during that time, our team will collect all the votes and identify the 1 winner out of the 3 groups that are going to be presenting now. So without further comments, we'll start with the first presentation. And the first presentation is by a group of researchers, Diana Laird from UCSF, Miguel Bueno Enriquez here from MWRI, and Melissa Holmes from the University of Toronto. And the title of their presentation is Using Interspecies Chimeras to Understand Female Reproductive Lifespan. So I'll invite the first group. Please come up.
Well, thank you. It's a real pleasure and an honor to be invited here and to be here in person as a finalist for the Magee Prize. So I'd like to start by telling you that it's obvious we're all aging. I've demonstrated this a bit at my own expense with the help of some AI. But the good news here, and it's okay to laugh, the good news here is that for those of us in the developed nations, such as the US and Canada and North America, we can expect, thanks to modern medicine and sanitation of recent years, we can expect to enjoy approximately twice the average lifespan of our forebears only 150 years ago. This is the good news. The bad news, however, is for those of us with ovaries, we can't expect to enjoy any additional reproductive lifespan. So to continue, I'd like to invite Miguel.
So what we want to know and we want to tell you today is why this is important. You've been hearing this morning about how aging could be affecting every single layer of our body. And it's also important to say that each tissue has their own time to do it. It could go to little places like the little wrinkles coming here, to other aspects of the biology that could be affecting the entire system. This will go to muscle atrophy, it could go to loss of bones, could go to an increased risk of cancer, but also could be affecting mental health. The important part here is that all these situations are affecting our population, and they are turning this into a public health issue. And rephrasing Sharon this morning, this is maybe one of the things that will make public health sexy back, right? Also, there's a lot of things about aging that we don't really know. But what is actually aging? When we think about aging, it's like when you buy a car, right? So you are expecting that everything in the car will be okay for 10 or 15 years. However, that is not happening. Some parts fall apart one day, other day. However, we know that now in biology, that could be tweaked. And this is based on two really insights of aging. The first one is that similar to what we can do with development, now in aging, we can also do genetic control of the lifespan. This information comes from 30 years ago, and we'll learn this lesson from a little animal, the worms. In 1993, Cynthia Kenyon analyzed one gene, and that gene is involved in the pathway of insulin. And she saw that when you take out the function of that gene, you are able to extend the lifespan of the worm in 30 days. That probably for all of you, 30 days like, duh. However, this means that it's expanding 2 times the lifespan of the worm. But also, with other studies published in 2003, it also supports the idea that these kind of pathways are really well conserved in the species. And in 2003, a mutation in mouse showed the same effect. And in fact, one thing that was really important about that experiment is that in the case of the female, the extension of the lifespan was even longer than in males. So we can modify aging, modifying genetics. We can extend the lifespan. The second important thing here is aging could be reversible. Everybody probably here wants to be younger, right? So we have examples in the literature that they support the idea that we can do this. And one of the examples of this is the young blood. In scientific terms, it's the parabiosis. And what are we doing is that blood from a young animal is affecting an old one and improving how the muscle or the brain is working. But we are here to talk about reproduction, right? We start with the fact that the ovary is not doing this. And the big question here, or one of our big questions is like, okay, so are you able to rejuvenate ovaries? And the answer is maybe. Biology could be a little bit complicated for females. And it comes from the base that the total number of oocytes or eggs that the female mammal will have, they are created in utero, or almost for all the mammals. So, thinking in that, we know that with age, it's not just the fact that we have less oocytes. It's also that the quality of those oocytes or eggs are dropping, and that increases the risk of miscarriages and also aneuploidies. One important thing is that this phenomenon is really well conserved between the species. However, the chronology of them is different. Because, for example, in the case of the mouse, this will be around 1 year, meanwhile in human goes for a little bit longer. So the big question here is how chronology and how clock could be tweaked. And to talk about how we can do that, Melissa will explain to you our big picture.
Thank you very much, Miguel. So, essentially what we are doing is we are proposing to marry the two major insights we have had into aging. From comparative genomics, we've learned that in some species, the clock can be slowed down. And parabiosis has taught us that we can take young blood, like blood from a young animal, and essentially turn back the clock or rejuvenate an older individual. So we hypothesize that we can slow the clock by questioning whether we can take cells from a long-lived animal and have those cells essentially teach the cells from a shorter-lived animal how to age better. The question is which species will be the teacher? So, many of the answers to medicine's biggest questions are already out there. In terms of aging, evolution has already solved how to make species live long and live well. So we've made a graph here to highlight just a very brief sliver of how variable maximum lifespan can be among mammalian species, including rodents, whales, and primates, including humans. So most animals, their reproductive lifespan will mirror their total lifespan. What that means is that they will reproduce for their entire lives. Humans are a very notable exception to this idea. So by capitalizing on the diversity of mammals, we think we can select the right species to teach us how to slow the clock. And we've done just that. So it is my tremendous privilege and pleasure to introduce you to the naked mole rat. Thank you. This one's from my lab. So I have had the pleasure of working with naked mole rats for over 15 years, and I say with absolute confidence that they are revolutionizing biomedical research. These animals live in large, complex social groups with a social hierarchy and in which individual animals take on different social roles, similar to what you would know of with bees, and quite frankly, not dissimilar to how humans work. They are small. They're approximately mouse-sized, which makes them incredibly amenable to laboratory work, and they are the longest living rodent ever identified. So they live over 30 years. This is 10 times longer than what you would expect for an animal of this size. Okay. Now, what's important for the purposes of our work is that not only do they live so long, they age so well. They are incredibly resistant to cancer, and they show no evidence of reproductive aging. So a 30-year-old naked mole rat will produce the same number of offspring as a 1-year-old naked mole rat. Thus, this is the ideal species to teach us about how to slow reproductive aging. So, the ovary is important to reproductive aging, but it does not work in isolation. The ovary is part of a 3-tiered system, a brain-body axis, if you will. And it is called the hypothalamic pituitary gonadal axis. The hypothalamus in the brain is actually the master controller of this system, and it sends signals to stimulate the pituitary gland, which in turn releases hormones into your bloodstream, which then make their way down to your ovary to stimulate the ovary to produce and release classic reproductive hormones like estradiol and progesterone, and also that stimulate follicular maturation and release. So, important for our work is that this axis is conserved in every vertebrate species. It is one of the most highly conserved brain-body systems across evolution. There we go. So using cutting-edge molecular techniques, we can examine this system at a previously unimaginable level of resolution and precision. So we will use single-cell multi-omics to investigate the transcriptomic, that is to say the gene expression profile, as well as the epigenetic processing of each of these tissue types. So the hypothalamus, the pituitary, and the ovary. By using these tissue, or by identifying and profiling these tissue types separately, we will create tissue-specific cell atlases, and by using bioinformatics, we will create the cellular and molecular networks that reveal how these tissues and cells work independently and how importantly they work together at both a tissue level and a systems level. Now, in addition to the tissue level profiling, we will make 3 additional comparisons. We will compare young animals to old animals, we will compare mice to mole rats, and we will capitalize on the remarkable social structure of naked mole rats to compare age-matched animals that are reproductive or reproductively subordinate. And this allows us to tease apart the effects of age versus reproduction. So collectively, this work will tell us about the remarkable cellular and molecular adaptations in naked mole rats and about the respective effects of age versus reproduction. And then finally, this work will produce an unbelievable amount of data. We will make these data freely available and open access, and this creates an opportunity for scientists around the world to mine these data and enabling discovery and have impact that goes far beyond our own work. Thank you. Diana.
Thank you, Melissa. So for the second part of our study, our aim is to resolve the individual contributions of cells within the HPG axis to reproductive aging. And importantly, we'll do this with a new platform that we've developed, which we call interspecies complementation chimeras. Now, a chimera and an interspecies chimera is an animal, a single animal, which is comprised of the cells of 2 different species. And the way that we will do this is starting with the stem cells, pluripotent stem cells from naked mole rats, which we will generate from a reproductive queen by reprogramming. These cells will then be introduced into an early mouse embryo. First of all, so that these cells can develop an organ or a cell lineage of choice from the get-go. The second reason that we introduce the cells at such an early stage is to avoid immune rejection. But we want to direct the development and the differentiation of these cells, so we have a special genetic trick, which is that using, in our case, an oocyte-specific promoter, we can drive expression of pre-recombinase, which then leads to the activation of a diphtheria toxin fragment, which will surgically and neatly delete the oocyte precursors in the mouse embryo. So this will develop a mouse with no germ cells, but importantly will open up a niche for those naked mole rat stem cells to repopulate, since they're less competitive than the mouse. And importantly, as we then transfer this embryo into the uterus of a recipient mouse, we can grow up a mouse which has the oocytes of a naked mole rat. And we'll use a similar strategy to grow up a mouse by deleting an early transcription factor in the pituitary. We'll produce a mouse with a pituitary chimerism, and finally, we'll produce a mouse with chimerism in the ovary as well as the hypothalamus. Now, in combination and separately, we can analyze these different chimeras, and using the single cell atlas that was described to you by Melissa, we'll understand what properties were changed. But importantly, what are our expected results? First of all, based on the function of the pituitary, we don't necessarily expect the pituitary chimera to influence reproductive aging. However, we do expect that chimeras in the hypothalamus and ovary support cells will alter the chronology, and we think that this may be based on altered signaling through the AMH or the FOXO pathway. We're also interested in the oocyte chimera, as we think that there may be enhanced survival of oocytes in this chimera based on perhaps increased ability to repair DNA or other insults in the naked mole rat oocytes. And an important question from our studies is whether a mouse environment will change the chromatin landscape in these cells in a mouse systemic milieu, and this is what the ATAC-seq will tell us. But what I want to convey to you is that this platform is entirely new and is quite versatile, so that we will not only be able to make discoveries in the field about reproduction, but in theory we can delete any cell line of any cell lineage desired to make a chimera. So I just want to make it, for a moment, tell you about the team. We've assembled an international team of experts. Melissa Holmes in Toronto has built what rivals to be the largest colony of naked mole rats in North America. She will supply the tissues. Those tissues are sent to San Francisco to my lab, where we will produce the chimeras, reprogram the stem cells based on our expertise in interspecies chimeras and mouse genetics. Once the chimeras are produced, the ovaries will be sent to Pittsburgh, where Miguel is one of the foremost experts on the ovary of naked mole rats. He recently published a paper, which is the first paper on the naked mole rat ovary in 50 years. And finally, the pituitary and hypothalamus will be sent to Toronto to Melissa's lab based on her expertise in neuroendocrinology of naked mole rats. And it just goes to mention that we also have collaborators who can bring expertise in the bioinformatics, in stem cell reprogramming, and in creating the complementation chimeras. So I think we have assembled a really great team, and I'd like to give our team the prize for having the most fun together in addition. So what's the impact of all of this? If I can advance my slide. I've told you that our project is to create a discovery platform, which we think will form the basis for identifying pathways which can be modulated by drugs, and also by evaluating and even prioritizing different interventions that can extend reproductive aging for women. But if we're modulating reproductive aging, are we possibly affecting systemic aging? And this is where we know that there's a deep evolutionary link between reproduction and lifespan. And numerous epidemiologic studies in humans suggest that after menopause, a woman's risk for a number of diseases of aging, including metabolic disease, cardiac disease, and neurologic disease, all of those risks increase. So is it possible that by turning back the reproductive clock, we may be able to promote healthier aging in women everywhere? That's our hope. So just in closing, I'd like to thank you for attention. I'd also like to pay homage to the talented trainees in all three of our labs, the ones who really put the long hours in front of the bench, in front of the computer terminal, and who really are the future of biomedical sciences. Thank you.
I just want to say, I really hope you stay forever young. Our next group, again alphabetically, is the group of Pam O'Ali here from MWRI, Kyle Orwig, also here from MWRI, and Caroline Gargett from from Monash University in Australia. And their title is Vaginal Stem Cells, the Missing Link in Vaginal Reconstruction. And Caroline's going to join us. I can see her already here by video. Thanks for joining from, I guess it's pretty late at night, huh? Or in the morning again?
Early morning.
Early morning, thanks.
All right, we wanted to start by thanking the scientific committee for the opportunity to present this proposal. And this is the tricky part, huh? The vagina is central to female sexuality, her sexual health, body image, and a sense of well-being. And at the most fundamental level, the cellular composition of the vagina drives its functional properties. Most cells of the vagina are derived from the epithelium and mesenchyme. The epithelial cells are arranged in folds or rugae to afford the property of distension that is needed for vaginal birth. The mesenchyme is divided into the subepithelium, the muscularis, and the adventitia. The cells of the subepithelium, a bit tricky, make structural proteins like collagen and elastin that give the vagina its mechanical properties. The smooth muscle cells of the muscularis are arranged in bundles that are oriented in multiple directions that afford the vagina its unique contractile properties. The cells of the adventitia provide a mechanism for communication with adjacent pelvic organs. Unfortunately, all or a significant portion of the vagina is physically absent in 1 in 5,000 girls at birth, and in tens of thousands of women who undergo surgeries for the definitive or life-saving treatment of pelvic organ cancers. In those whose vagina is physically present, functional loss can occur as a result of radiation, chemotherapy, menopause, and aging, especially in those women and girls who can't use hormones. In those experiencing a physical loss, current therapies to create a new vagina involve the use of grafts derived typically from skin or intestine. And complications after these procedures are common. These tissues are derived from different cells. Their cellular composition is different, and therefore they don't have the appropriate functional properties. Because they're placed in a foreign environment, they tend to scar and narrow. Those derived from skin are plagued by hair growth, while those derived from bowel are associated with a copious discharge that can be irritating. And finally, because these tissues are derived from affected women, they are associated with significant morbidity at the donor site. For those girls and women experiencing a functional loss, lubricants are offered to provide moisture. However, these agents are short-lived, cytotoxic, and they can alter the pH, increasing risk for infection. Over the past decade, lasers have been introduced into the market. These devices are designed to superficially injure the vagina to promote a repair response. However, they can be painful, they're expensive, and recent multi-center randomized trials have shown that they're not clinically effective. So current therapies to restore vaginal anatomy, structure, and function are suboptimal, leaving affected girls and women with few good options.
We propose that vaginal stem cells have the highest capacity for improving therapeutic surgeries and therapies aimed at reconstructing the human vagina, and most importantly, restoring vaginal function. Now, to do this, we first need to consider how stem cells function, which is primarily regulated by their stem cell niche. It's a specialized microenvironment which determines stem cell fate decisions, whether to stay quiescent, whether to proliferate, or to differentiate. Niches involve a blood vessel, which provide the niche cells, including pericytes and adjacent stromal cells. Tissue damage triggers stem cell function. It is sensed by the vascular cells, which signal to the stem cell to undergo an asymmetric cell division to produce an identical copy of itself and a more differentiated progenitor, which goes down to produce transit amplifying daughter cells, which rapidly expand and differentiate into the functional cells of the tissue, thereby replacing those lost cells and affecting tissue repair. There's strong physiological evidence that the vagina is a stem cell-based system. It undergoes dramatic structural and functional changes in response to ovarian hormones over the life cycle. For example, there's cyclical changes during the menstrual cycle. There's dramatic thickening in pregnancy, and there's tremendous capacity for healing following vaginal childbirth-induced trauma. This led us to hypothesize that the human vagina is a stem cell-based system comprised of epithelial stem cells, mesenchymal stem cells, unknown stem cells that maintain vaginal tissue organization and function.
Well, it's my honor to be able to introduce you to our multidisciplinary and collaborative team. We're led by Dr. Pam Moali, who is a successful physician scientist and also a reconstructive pelvic surgeon here at Magee. And she treats the women and girls with vaginal dysfunction who are the focus of our proposal. I'm Kyle Orwig, also at Magee. I'm a developmental and reproductive biologist, and my area of expertise is stem cell transplantation and regenerative medicine. And finally, Pam and I are joined by Carolyn Gargett from the Hudson Institute in Australia, who is a pioneer in the study of stem cells in the uterine endometrium, and she'll be able to bring that experience to bear in our studies of the human vagina. So, For AIM-1, our objective is to discover and define vaginal stem cells that exist in the vagina, as well as those essential niche cells that support vaginal stem cell function, using unbiased, high-throughput, single-cell transcriptomics. Well, as you can see in this image, the vagina is a complex tissue. It's made up of several individual cell layers, each of which is made up of many different cell types. And as Carolyn explained, we know that the vagina is a stem cell-based system due to the regenerative properties that we can observe through the life course of a woman. But we don't formally know which one of those cells is the stem cell or the lineages that they produce, and we know virtually nothing about the vaginal niches that support stem cell function. So, to address this knowledge gap, we will obtain human vaginal tissues that we will digest with enzymes to produce a suspension of cells that we will then run through a 10x microfluidics device and perform high throughput single cell RNA sequencing of the vaginal tissues so that we can learn about the gene expression profile of each individual cell of the vagina. And then, using dimensionality reduction algorithms and cell clustering, we can identify cells in the vagina who have a gene expression profile that is more like each other than it is like every other cell type in the vagina. And using this unbiased approach, we'll be able to identify all the different cell types of the vagina. Well, we don't have data for vaginal tissue yet, but here I'm showing you data that we produced after single-cell RNA sequencing of the testis. This is work that was done by Sarah Munyoki, who is a PhD student in the laboratory, and her studies revealed the cells that comprise the stem and progenitor population of the testis. They also defined the spermatogenic lineage that arises from the stem and progenitor cells and ultimately culminates in the production of terminally differentiated sperm. And finally, her studies identified several other cell populations in the testis that define the stem cell niche and regulate stem cell self-renewal and differentiation. So in our studies, we imagined that we can use the same type of approach to understand the impacts of diseases, medical treatments, or injury on the cell types that exist in the vagina or the genes that they express. And we imagined that we can use these data to identify markers of each individual cell type of the vagina that then we will follow up with validation studies such as immunolocalization to determine where each of those cell types resides within the vaginal tissue. We expect that we'll identify cell surface markers that will allow us to isolate and enrich each of the different cell types in the vagina. And that would include putative stem cell populations that we will test functionally in vitro assays that will be described by Carolyn in AIM-2, and in vivo assays that will be described by Pam in AIM-3.
So in AIM-2, we aim to identify vaginal stem cell populations using candidate stem cell markers previously identified in other organs. First, we need to immunostain for these candidate markers to determine that their presence in human vaginal tissue. For the epithelium, we have selected CD49F, the most commonly used epithelial stem cell marker. And here in human vagina, CD49F stains cells of the basal epithelial layer as shown in green. We will combine that marker with other markers to more precisely pinpoint the stem progenitor cell population in that basal layer. For the mesenchymal stem cells, we will use SUSTI2, which is a marker of bone marrow, endometrial and placental mesenchymal stem cells. Here in human vagina, it stains the perivascular cells throughout the mesenchymal layer. And in particular, there's a network of SUSTI2 positive vessels under the basal layer of the epithelium, and we predict that those SUSTI2 positive cells are niche cells for the epithelial stem cell. And similarly, we will undertake immunofluorescence staining on human vagina for surface markers of novel stem cells identified in AIM-1. Now, having determined that identified candidate stem cell markers are present in human vagina, we then assess their utility in vitro functional assays. So for the epithelial stem cells, we will separate the epithelium from the vaginal tissue, dissociate into single cells, and we've shown by flow cytometry that the epithelium contains 5.4% of CD49F positive cells. By our screening assay, stem cell screening assay or clonogenic assay, CFU assay, we've demonstrated that these form epithelial clones. We will also undertake more stringent stem cell assays such as self-renewal, proliferative potential, and differentiation by using vaginal epithelial organoids. Similarly, for the mesenchymal stem cells, we will dissociate the mesenchymal layer, we have shown already that that mesenchymal layer contains 3.2% SUST2 positive cells. We will sort both the SUST2 positive and the SUST2 negative cells and assay them in our CFU assay as we've done previously for endometrium. And we're predicting that the SUST2 positive vaginal cells will be the clonogenic cells of human vagina. We will then test their mesodermal lineage differentiation and the other stem cell assays in vitro, and thereby verifying the MSC properties of the sorted cell populations. These assays will enable us to select the most appropriate candidate and or novel markers identified in AIM-1 of vaginal epithelial stem cells and mesenchymal stem cells from human vagina for assessing in vivo in AIM-3.
The overall goal of AIM-3 is to assess vaginal stem cell function in vivo by demonstrating that depletion of these cells by systemic chemotherapy creates a space for engraftment of donor cells in recipient niches. As our donor cells will use vaginal stem cells isolated from young women, as our recipients will use stem cell depleted nude mice. And we hope to develop this as a novel bioassay for use in future studies evaluating the regenerative potential of vaginal stem cells after depletion by a variety of other therapies. So for our experimental approach, we will treat nude mice with chemotherapy, establishing a regimen that will deplete both stem and niche cells. For our donor cells, we will take full-thickness vaginal biopsies and isolate out vaginal epithelial stem cells, mesenchymal stem cells, and a single cell suspension, as well as novel cell niche cells identified in AIM-1. And our interest in these two latter cell populations is to test their capacity as niche cells. We will then transplant vaginal epithelial stem cells alone, epithelial stem cells plus mesenchymal stem cells, or all three cell populations, into our nude mice 6 weeks after chemotherapy. 8 weeks later, we'll test the stem cells for proliferation, engraftment, and restoration of vaginal structure and function. And as mentioned previously, we will develop this as a novel bioassay in which we test the capacity of human vaginal stem cells to restore vaginal structure and function after a variety of other treatments, including multiple chemotherapy regimens, radiation, and conditions like aging and menopause.
So to conclude, we will use a combination of novel, state-of-the-art, and traditional approaches to establish and validate a technical platform for studying the human vagina.
In testing the stemness of our novel stem cell populations, we develop a novel bioassay to test stem cell survivability, proliferation, engraftment, and restoration of vaginal function after transplantation.
By defining human vaginal stem cells in their niches, we will fill a major knowledge gap in our understanding of vaginal structure and function, which in turn will lead to the development of novel therapies and provide hope for affected women and girls. And we'd like to thank all of the faculty, trainees, and staff who contributed to this presentation today, and we are particularly grateful to the Richard King Mellon Foundation and the Magee-Womens Research Institute for providing us with this funding opportunity. Thank you so much.
Great. Our third presentation, again alphabetically, is by a group of researchers. The PI is Soman Paul from University of Kansas Medical Center, accompanied by Melissa Mann here from Magee-Womens Research Institute and William Pastor from McGill University in Canada. And their title is Epigenetic Regulation of Trophoblast Function, Infertility, and early pregnancy loss.
So our team is united in our goal to understand the molecular mechanisms that lead to infertility and early pregnancy loss. Mammals are different from all other species, which lay eggs in which their young develop. Mammals are different from other animals because they house their offspring in their bodies and grow them. To carry out this adaptive function, mammals have developed a unique and specialized set of cells called the trophoblast cells. These cells, which are depicted in yellow, we're showing both the mouse and the human, where the embryo is fertilized and the oocyte is fertilized and the embryo develops. And the yellow cells are the outer cells of the embryo, shown in both mouse and human. And these cells are essential for anchoring the embryo in the mother's uterus. So here's another schematic of the early embryo, and there are different types of cells. The embryonic cells are going to give rise to the entire fetus, where the yellow trophoblast cells are going to give rise to the placenta. The placenta is an essential organ required for healthy pregnancy. And that's because the trophoblast cells will develop into the placenta that are going to carry out the function of many different organ systems until the embryo is ready to take over. This includes the lungs, which carries out the exchange of oxygen for carbon dioxide. It acts like the stomach and the intestines for nutrient uptake and delivery. It acts like the liver and the brain for carrying out hormone production and secretion. The kidney function removes the waste, and the epithelial system must regulate temperature as well as immune function. So the placenta is an extremely important organ. However, when there is impairment of this development or impairment of the function, we can see that there is infertility, implantation failure, as well as pregnancy loss. These are conditions that are devastating for the patient and their partners, as well as for the clinicians. If we look at all conceptions in the human, 30% are lost before the embryo makes contact with the mother's uterus. 30% more of pregnancies are lost before 6 weeks of age. And another 10% are lost as early miscarriages up to 6 to 22 weeks. Another way to state this is that 1 in 4 women experience pregnancy loss, and 1 in 100 women experience recurrent pregnancy loss, which is defined as 3 or more pregnancies. For these conditions, 50% of these losses are unexplained. Now, the risk factors that are associated with infertility and early pregnancy loss include those that are biological, like genetic reasons where there's chromosomal abnormalities, uterine, where there may be abnormalities in the uterine structure, and then there are lifestyle issues, which could increase the chance of implantation or pregnancy loss, and they include things such as alcohol, obesity, and drugs. When we have these two factors together of a genetic component and an environmental component, this is an arrow to suggest that there may be epigenetic reasons that are contributing to early pregnancy loss. Now, epigenetics simply means epi, on top of, and genetics is the genes that carry out the blueprint of the body. So the epigenetics can be viewed as the conductor, which directs genes. When the genes are open, the conductor says the genes may be turned off. Whereas when the chemical reactions cause the chromatin to be really compacted, it tells the genes to be turned off. So I'm going to turn this over now to My teammate, this is Dr. Schumann Paul, who is an expert in trophoblast and early trophoblast development.
Thank you. And you just know about the terminology epigenetics. And this is very important in the context of trophoblast cells and the placenta, because an embryo needs to grow, and at the same time the placenta needs to grow. So that means the progenitor cells needs to self-renew. At the same time, the developing placenta needs to mediate functions which Melissa just explained. It functions like multiple organs. So it needs to develop specialized cell types which mediates different functions. So how the same gene content in different cells are leading to different behavioral patterns, different functions. So that means those genes are either turned off selectively or turned on selectively in trophoblast cells, thereby ensuring the self-renewal of progenitor, which ensures the development bigger placenta, or formation of the specialized trophoblast cells. That means they can mediate the functions that is needed for nurturing or developing the fetus within mom's uterine environment. So I would like to tell a small story. One of my friends, very close friends, are suffering from 6 recurrent pregnancy losses. And while we were talking in a party, she asked me, hey, Shoman, you are a stem cell biologist, developmental biologist, can you look into it? I thought, okay, this is an opportunity. And that drove me into this project in 2018. And I thought, okay, let's look at the placenta from a normal sporadic miscarriage versus a recurrent pregnancy loss. And when we looked at collected placenta from pregnancies which are associated with recurrent pregnancy losses or some sporadic miscarriages, we saw extensively differences in some RPL associated or recurrent pregnancy associated placenta. You can see on the top image that a normal placenta, when it grows, it forms a villus, finger-like structure. And in some of the recurrent pregnancy losses, those structures are not there. So it shows there are a fundamental difference within the placenta that could lead to these recurrent pregnancy losses in these patients. So what we did, we took those placenta and we just looked the gene expression pattern in an unbiased fashion. And what it resulted to show that one of those genes which is altered in recurrent pregnancy losses is KDM1A. KDM1A is also known as lysine demethylase A, is the first identified histone demethylase protein. So this protein, it takes out a methyl group from a histone which wraps the DNA and makes it condensed state. So we thought that KDM1A alteration might be important for the placentation process. And that's what we looked at the literature and the biology in another mammalian system, which is mouse. So the advantage of mouse is, unlike in human, we can delete a gene in mouse and study them. So the KDM1A deleted or knockout mouse showed that the developing placenta, the extraembryonic tissue, is absent when the KDM1A gene is deleted from a mouse embryo. And those embryos die at E7.5, which is equivalent to a stage which is the first trimester of human pregnancy. We also noticed that pre-implantation embryo, those embryo which has not attached to the mom yet, if we delete that gene, KDM1A, from a mouse embryo, we saw implantation efficiency is altered. So you can see in that cartoon as a normal uterine horn of a mouse with a lot of different bubbles, those are all growing babies. But if KDM1A gene is deleted from a pre-implantation embryo and then we transfer to mom's uterine horn, we can see those babies are not developing because of implantation failure, and some of those babies, they are not forming properly because their development is impacted. So from these studies, from 3-pronged analysis with recurrent pregnancy losses, placenta, the mouse models, as well as the implantation failure, led to our hypothesis and our project here. So our hypothesis is that KDM1A could be the conductor of trophoblast epigenome. and it orchestrates the gene expression pattern in trophoblast progenitor cells so that they can self-renew, placenta can grow, and also it can potentiate those progenitor cells to differentiate into specialized cells so a developing placenta can function properly and can mediate the necessary functions that allows the pregnancy to go on and form a healthy baby. So what we did, as I said, as Melissa said, evolution has selected mammals to grow their baby inside their body. That's the advantage of the mammalian system. So we are going to utilize that. So as we cannot generate a genetically knockout or genetically modified human, we can use a mouse model to study the pre-implantation development and test the implantation efficiency and the pre-implantation developmental stages. So we are going to use a mouse model where we'll conditionally delete the KDM1A gene in pre-implantation embryos. We'll look at their implantation efficiency, formation of trophoblast stem cells, and as well as we'll also look at their biology in multiple contexts, especially in the context of gene expression. We cannot also generate a human mutant to study post-implantation placenta, but in 2019, there's a huge advancement in the field that first human trophoblast stem cells are derived in Japan. So, and that is a timely discovery, so we thought our laboratory took that opportunity, and as the first laboratory in the United States or in the world, we first generated the patient-specific trophoblast stem cells from placenta which are associated with recurrent pregnancy losses. So this huge advancement of generating patient-specific stem cells given us an opportunity to test whether recurrent pregnancy losses are in generally associated with defective KDM1A function, and whether the loss of KDM1A function is affecting the self-renewal process or their differentiation to specialized trophoblast cells. So these are the two innovative systems we are going to utilize to execute our research. And our specific aims will be described by our another team member, Will Pastor. He is a stem cell biologist and working in the McGill University in Canada. And I'm so happy to have Will over here. Thank you.
All right. In order to study these biological systems, we're going to use a number of cutting-edge technologies. These include nucleosome occupancy and DNA methylation sequencing, which can be used to determine the epigenetic state of a cell. Single-cell accessible chromatin sequencing, which can tell you what genes are capable of being turned on in a given cell, as well as single-cell RNA sequencing, which tells you what genes are on in the cell at any given time. So together, these technologies can tell us the epigenetic state and the transcriptional state of all the different cell types of the placenta. Finally, we use a literal cutting-edge technology, CRISPR-Cas9 gene editing, which allows us to mutate or reduce the expression of KDM1A. Now, we have pretty clear evidence that KDM1A is implicated in normal trophectoderm development and in recurrent pregnancy loss, and what we want to find out is mechanistically what is it doing, and we're going to use two different experimental systems. First, we're going to look at mice. We already know that mice that lack KDM1A fail to form trophectoderm correctly, so we're going to look earlier and earlier at mouse development to figure out exactly where the KDM1A deficient mice go wrong, and using the techniques I talked about in the previous slide, how their epigenome is malfunctioning as a result of loss of KDM1A. Second, we'll work with stem cell models. Stem cells basically do two things. They divide indefinitely to grow the organ, to grow the placenta, and they differentiate to form the different cell types of the placenta. We hypothesize that KDM1A is going to be important for either self-renewal or differentiation of placental stem cells, which is something we can model in vitro using trophoblast stem cells where we've mutated KDM1A, or using trophoblast stem cells from recurrent pregnancy loss, which naturally express low levels of KDM1A. So we'll be able to figure out what's wrong with the stem cells and how this contributes to the phenotype observed in recurrent pregnancy loss placenta. The outputs of our project, in the narrow sense, we're going to figure out mechanistically what KDM1A does to form healthy placenta, and how it's implicated in recurrent pregnancy loss. In the long run, we're trying to open a new research program to understand the molecular basis for this otherwise very poorly understood phenomenon of recurrent miscarriage. I would like to thank the Richard King Mellon Foundation and the Magee-Womens Research Institute and Foundation for inviting us to this wonderful conference. The talks have been great so far. I think we've had 3 very good proposals.
Thank you so much.